Autism Spectrum Disorder, also known as ASD, is a lifelong developmental condition that impacts the way an individual interacts with both people and the environment. The reason autism is defined through a spectrum is because individuals with the disorder are affected in different ways. Some are categorized as high functioning individuals, while others are considered as low functioning and require significant support. The fifth edition of the Diagnostic and Statistical Manual of Mental Health Disorders, also known as DSM-5, requires abnormalities in two main areas to clinically diagnose ASD. The first criteria revolves around deficits in social communication and interaction. This can manifest through behaviors such as nonverbal communication, inability to engage in conversation or maintain eye contact, and difficulty maintaining relationships. The second criteria focuses on the presence of restricted or repetitive behavioral patterns. This can manifest through behaviors such as an inflexible adherence to routine, an extreme distress to small change, fixated interest in unusual objects, and oversensitivity to sensory stimulus. This video will discuss the neurological basis for the behaviors associated with these two criteria, specifically focusing on abnormalities in both the ratio of neurotransmitters within the brain as well as the overall neural networking across the brain. Neural networking can be described as the wiring of the brain that allows communication between different cortical and subcortical areas of the brain. This process is achieved through the firing of signals, known as electrical impulses, by the cells of the brain, known as neurons. When looking at autistic individuals, multiple anatomical studies have actually shown an excess in the number of neurons or brain cells found within the critical parts of the brain. This excess is particularly evident in the temporal and frontal regions of the brain, where an overabundance of 60% in brain cells were found in autistic individuals when comparing to controls. Anatomical studies have also noted a general increase in the overall brain volume among individuals with autism spectrum disorder or ASD, where this increase has been attributed to the abnormal neuronal growth during gestation. The abnormal abundance of neurons in the autistic brain have been linked to miswirings within the brain, where the firing of electrical signals between neurons are substantially mismanaged, resulting in low levels of neural connectivity or networking. This understanding has been particularly evident between the posterior regions of the brain to the prefrontal cortex, where an excess number of neurons within these areas have caused errors in neural connections. These neural connections within the prefrontal cortex are vital in helping individuals learn social and communication functions. Thus, disruptions within these areas can directly manifest the abnormal social behaviors described by the DSM-5 criteria mentioned earlier. Moreover, other anatomical studies have identified that individuals with autism tend to have more white matter in the brain than controls, with approximately 18% more neurons found within the cortical areas and 39% more neurons in the cerebellum. An increase in white matter within the temporal cortex could explain recent findings of low neural connectivity within the fusiform gyrus found in this cortical region. The fusiform gyrus is the area of the brain ordinarily used for facial recognition. Researchers suspect low connectivity within this area to be linked with why some autistic individuals find it difficult to recognize faces. Further studies within this area have found that in order for autistic individuals to compensate for the low connectivity in this brain region, individuals with ASD use the inferior temporal gyrus instead. This region is normally reserved for processing objects and therefore means that individuals with ASD process faces in the same way they process any other ordinary object, making it difficult for them to distinguish unique features and link those distinct features to a specific person. As poor facial recognition can negatively impact the forming and maintenance of social relationships, the presence of low brain connectivity further categorizes social behaviors highlighted by the DSM-5 criteria for autism. There has also been research to suggest that individuals with ASD have an increase in the ratio between excitation and inhibition neurotransmitters and receptors. This means there are more excitation mechanisms in the autistic brain than inhibitory mechanisms. This is because there are reduced GABA receptors and abnormal glutamatergic and GABAergic neurotransmission in the key regions such as the neocortex, hippocampus, amygdala, and cerebellum. The decreased inhibitory mechanisms in the brain, combined with poor connectivity, has been known to directly impact the inhibitory control of those with ASD, often resulting in repetitive behavior and an inability to restrict emotional outbreaks. It should be noted that studies used to draw these conclusions vastly differ in participants. Looking at autism as a spectrum, behavioral studies often use patients with less severe forms of ASD, as those with severe ASD usually have a range of other co occurring disorders that could limit their observable behavior. Contrastingly, anatomical studies that give insight into the brain structure have mainly used low functioning individuals. These differences thereby limit the reliability of concluded links between the brain and behavior within current ASD research, as they do not consider the entire spectrum. To conclude, the neurobasis of autism can largely be attributed to a larger brain volume, poor brain connectivity between different regions, increased white matter, and decreased inhibitory mechanisms within the brain. Further research needs to be conducted to add reliability and credibility to these results and findings. Thank you for your time, and we hope you enjoyed this presentation.